Okay, so it doesn't really look like anyone else is going to show up. Oh, never mind. I'll just admit Cawthor. Good morning, Cawthor. All right, so I'll talk about recitation four. I'll go through the PowerPoint real quick, just so anyone who might have questions on what I present and aren't able to attend office hours can actually watch this recording. Um, so of course we're looking at gene interaction or non-Mendelian inheritance patterns, and specifically we wanted to look at dog colorings. So how the coat, both the coloring and the characteristics of the coat may follow gene interaction or non-Mendelian inheritance patterns. So, of course, with the learning objectives for this recitation, you'll be able to understand limitations of basic Mendelian inheritance patterns. As you can imagine, for more complex animals, even for dogs, there are multiple genes that go into some of the phenotypes that you see, such as coat color. Through this PowerPoint, you should be able to understand epistasis, incomplete dominance, codominance, and modifier genes, and then apply those principles to the genetics of coat or dog coats. As you'll see, some of the questions in the recitation quiz go over dog coats as well as some other phenotypes in plants. So just going over Mendelian genetics, we've pretty much just been talking about phenotypes, which are the physical traits, and genotypes, the genetic composition from the Mendelian standpoint, such as the simple Punnett squares and one gene directly affecting a phenotype. So this includes dominant and recessive alleles, Mendel's laws of segregation, and independent assortment. So these are really important ideas to genetics. But as I mentioned before, there are multiple genes that interact in order to give specific phenotypes. Epistasis, incomplete dominance, codominance, and modifier genes are all examples of these non-Mendelian genetics. So if we were to use Mendelian genetics on a dihybrid cross, and the dihybrid means that both of the individuals that are crossing are heterozygous for both genes A and B in this example, we would get this 9 to 3 to 3 to 1 ratio in a 4 by 4 Punnett square. So as you can see here, there are 9 individuals that are denoted by these white boxes, and that's because they are either heterozygous for gene A and or gene B, but these three yellow boxes are all homozygous recessive for gene A, while not gene B. And then finally, this final box is homozygous recessive for both genes or alleles A and B. So whenever you don't get this 9 to 3 to 3 to 1 ratio, you're going to notice that Mendelian genetics is not used, and rather there might be some gene interaction at play. So some of those gene interactions can include epistasis or one gene masking another. So I hope this makes some sense in that epistasis is the phenomenon where the effect of a gene mutation on one allele is dependent on the presence or absence of mutations on other genes. So the um, one hallmark example is dog coat coloring because you have this TYRP1 gene at locus B that gives a black coat when the dominant allele is expressed, and then a brown coat when the recessive allele is expressed. However, in order for the pigmentation to actually deposit into the coat, that's dependent on a different gene, MC1R, at locus E. So if the dogs are homozygous recessive for uh, both alleles on gene E or at locus E, then they won't be able to actually express the black or brown coloring, making them yellow. So you'll see here that uh, if the dogs have at least one copy of the dominant E allele, then they'll be able to express black or brown. But if they are homozygous recessive, it doesn't matter what's at locus B, you're always going to have a yellow coat. So the ratio that follows this type of epistasis, which is recessive epistasis, you'll have a 9 to 3 to 4 ratio. So 9 of the individuals will have a black coat, 3 will have brown, and then finally 4 will have a yellow coat. Yes, the recording will be placed in the Module 3 recording section. Um, if you do have to leave. So, as you can see, though, the 9 to 3 to 4 ratio is much different than the 9 to 3 to 3 to 1. So, recessive epistasis leads to this ratio. On the flip side, though, with dominant epistasis, um, this is pretty similar, except for the gene that is epistatic over the other is dictated by the dominant allele. So in this case, gene B is epistatic over gene A when the individual has at least one dominant allele. So you could see here that all of the white colored, I think this is squash. I might like some type of squash. I might be wrong. But if there's at least one dominant B allele, you're always going to have white coloring. And that's similar to the dog coats in that the pigmentation, the yellow or the green, won't deposit onto the surface of the squash. Um, on the flip side, though, you'll have three squash in this 4 by 4 Punnett square that are homozygous recessive for B, but are have at least one dominant allele for gene A, which encodes for a yellow phenotype. And then finally, you'll have one squash that's homozygous recessive for both alleles. So that's the one green one. So once again, it's different from the Mendelian ratio. And then another form of gene interaction is incomplete dominance. So this is a little simpler than epistasis. You're not really worried about um, multiple genes, but these types of alleles are important because for incomplete dominance, you have what I like to call like a mixing of phenotypes. For example, poodles are usually homozygous dominant for this gene C, so they'll have super curly hair, whereas Labradors have straight hair. But if you um, mix these two breeds, you'll have a Labradoodle, which are adorable and actually have wavy hair, which is kind of like the middle ground of curly and straight. So then another example of gene interaction is codominance. And so instead of there being a mixing of phenotypes, you just have the expression of both phenotypes at the same time. Um, so you'll in this case, you have the white coat over here on the left and then the brown coat on the right. But if you mix them, both of the colors will be expressed at the same exact time. And now you're just going to have spotting. So then finally, modifier genes um, are genes that influence the phenotype given by another allele. So, for example, they can be plus or minus modifiers. So in Great Danes, the Harlequin modifier influences the color between the spots of the dog's coat. So even though the dog is spotted, there's an allele that has that, this gene modifies the actual color on the coat that's given by the spots or in between the spots. Um, so there's an example of that one. And then finally, some things that you're going to see on your recitation quiz are tomatoes' flesh colors. So you have this locus R that influences yellow versus red colored tomatoes. So red is the dominant, yellow is the recessive. But there's a second locus that actually turns that red into more of a green. So it modifies the red coloring. So if the tomato is homozygous recessive for the G alleles, then um, the tomato won't be colored red, it'll actually be colored green. So it's actually modifying the red coloring. And then also, human blood types are an example of codominance. There's going to be some questions about this as well. So there are two main alleles that influence blood types. The A and B alleles. They're just normally written as this, I to the A and then I to the B, while the recessive allele, little I, encodes for type O. So when both A and B are expressed, you have the expression of both alleles at the same time. You have blood type AB. And then obviously you see that IA and IB are dominant over the little I, because heterozygous individuals have either blood type A or blood type B. So then once again, I added these last couple slides in here just to remind you that this is an example of epistatic genes that uh, this recessive epistatic allele E will dictate whether a lab has a yellow coloring over a black or brown. And then making sure we understand that the distinction between incomplete dominance versus codominance, either the mixing of alleles or the expression of both alleles at the same time from different genes. And yeah, so those are the slides. So if you have any questions about anything on the slides, okay, Cawthor, have a good day. Um, you can ask questions about that, or if you have any questions about the quiz itself, feel free to ask away. So one thing that I will start off with, let me see here actually. Yes, so if you do have questions about activity four, you can ask those as well. Um, yeah, Brenda, if you don't have any questions about recitation four, we can also talk about activity four. I do just want to mention though that if you have not done recitation four yet, drawing out Punnett squares are very helpful. I'm really bad at visualizing Punnett squares. Um, okay, sweet. But yeah, I'm really bad at visualizing Punnett squares, and I always mix up my numbers in my head. So drawing them out might be really helpful, especially when you're doing something like a cross with two different genes. Um, but besides that, I feel like all of the other questions are pretty straightforward. Just feel free to rewatch either this recording or the recording of the PowerPoint itself if you might be confused about anything from any of these questions. I tried to address some of the different aspects in the questions so that they weren't too tricky, but yeah. So if we're all good with recitation four, yeah, we can look at activity four together. I'm going to stop sharing my screen there and then open up activity four. Okay. So this is the first time I'm actually looking at the worksheet itself. Um, I was able to hear from, I believe, we talked about this in one of our la previous TA meetings a little bit, but okay. So what questions might either of you have on activity four? Or we can kind of just walk through the whole thing together as well. Oh yeah, yes. You definitely are able to ask using your mic.
Okay, thanks, Luke.
Yeah, of course.
I was just wondering because I started it and the 3 point test cross, just like the 2.1, is supposed to be done using a tri hybrid cross with
Mhm.
since it's 3 point homozygous recessive at all three genes.
Right, right.
So that means that would be the answer to that first part A, right?
True. Yeah, I mean, I technically can't give the answers,
Oh
but
yeah, sorry.
no, 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 you're good. You're good. But honestly, like, yeah, you're
Because
that's definitely correct.
because the only thing I'm confused about is because then when it goes to C, based on the results above, the parental progeny, which means the offspring that have the parental like phenotypes. It says three: short, blue, and thin. But I was like, okay, neither of the parents have that as their phenotype. So I was just confused. So then I was like, okay, so if it's just going by that, then I'm like, then that means the other parental progeny would probably be seven. But um, I guess I was just confused about that. And then
Yeah. Okay.
doing like the double crossover. Uh, if I went by, so I did five and then four, and then if I use that, then it looks like the allele that changed between those two from the parental genotypes would be E. And then if I remember from module 3, I believe like the 3 point test cross analysis section, then that would mean that E would be in the middle.
I see what you're saying. Yeah, you're definitely on like the right thought process.
Hmm.
Going up to C real quick, when you're looking at the parental progeny,
Hmm.
when it asks about that, it's not really looking at, the phenotype itself, like whether that lines up to one parent or not. It's more just about how frequent the phenotypes in the progeny are.
Okay.
So even though it says like parental progeny, um,
Mhm.
since you can see that number three here, there's 500 progeny that are short, blue, and thin. The other one is going to also be the highest number. So that's going to be your 498 over here.
Okay.
Yeah.
That's
So
considered.
yeah, yeah, everything else that you said, I think that um yeah, you should be good on understanding, yeah, how to find the middle gene.
And then I guess because with that, with the pre-filled answers, then because it says to complete the figures below, and then for the remaining questions, oh, ignore the male gamete. I guess it just says rewrite the genotype of the mating results with the correct order. So does that mean that the pre-filled answers technically aren't correct, or was I the one that was off with that line of thinking that I was explaining?
Sorry, where did you actually see that sentence that you read?
Oh, let's see. Down below G.
Okay.
So it says like for H, rewrite the phenotypes with the genes in the correct order, and then in the red was the part about ignore the male gamete.
Oh, I see what you're saying here.
Which I mean makes sense because it's the tester one.
True. True. Yeah, so yeah, when it says ignore the male gametes, you just don't have to worry about the slash. Um,
Oh yeah, because it's always gonna be like the
the
recessive
yeah,
ones.
exactly. Yeah, all recessive, all lowercase. Of the mating results from the table. So to me, it seems like these pre-filled portions are already given to you. Let me double check on that though.
Yeah, they were on there, which is why I was confused because then I'm like, if my line of thinking that E was the one that changed, because I mean that's what I was seeing.
Right.
And then if I like move those like the alleles over the way they showed, then that would mean like E is in the middle, but then in those pre-filled ones, it's showing G as the one in the middle. So I guess that's where I was the most confused.
Definitely. Yeah, I see the confusion there. So I believe that those it's 5 through 8 that are already written. Yeah.
Yes.
You can leave those alone.
Mhm.
So you are just filling in those top four. So right, I can't. So yeah, unfortunately, I can't say whether gene E or
Yeah.
gene B is in the middle, but I think that looking at part H kind of gives a hint as to which one is actually in the middle.
Looking at part, which one?
H, like the way they're
H.
writing it there.
Hmm.
Yeah.
And I guess it since I mean it's true you can't give the answers, but could we take a look at then the process of um
Actually finding that middle gene.
yeah,
Yeah, definitely. So when you're looking at part C, they give you the parental progeny is going to be three, and then what would you say is the other parental progeny?
let's see. C it would be 3 and 7.
Right. So so they give you the geno or the genotype for three. So do you know what the genotype for seven would be? I don't know if you've ever
Yeah.
got that far.
Yeah, it would be, I'll just do it off the top. Capital E, lowercase b, um, capital G, and then slash all of the homozygous. So small e, small b, small g.
Perfect. Yeah. So that's good. So now that you have the parental progeny, the way to find the middle gene will be to look at the double crossover progeny phenotypes or genotypes. So I think you already mentioned that those are numbers 5 and 4, and that's just because um, they have like the lowest number of progeny. Um,
Yes.
okay, perfect. Does it make sense why it's supposed to be the lowest number?
Yeah.
Okay, perfect. So they give you the first genotype, little e, big B, big G. So what would the second phenotype or the genotype be for 4?
It would be big E, small b, small g.
Perfect. Okay, so in order to find the middle gene, you have to compare these two genotypes. So in order to do that,
Oh, okay. I see
yeah,
now. It's because I was still fixated on doing it off of like the original parental genotype.
okay,
Yeah, if we going off now, like with you explaining, like, okay, I should go off of like, the parental progeny from C, then okay, then it makes sense that it would be G in the middle, since that's the only one like comparing them, like with the genotype from 3 and then the genotype from 5, like G changed, and then from 7 and 4 comparing those. G's
right.
also that changed. Okay, that makes sense then.
Yeah, perfect. And then do you know if uh it matters whether you do like EGB or BGE? Like, do you know?
I think like if we, because I remember when they were only just using A, B, C,
Hmm.
and so like it since it already arranged it initially as EBG, I
Hmm.
think the way that it's supposed to be done is, let me see. Actually, yeah, no, I guess I don't know how they move G to the middle that way. So if you could explain that, sorry.
Gotcha. No, I mean, so we know that that's in like yeah, G's in the middle, but what's important is that since loci and like huge like big DNA sequences can be read left and right, the order of like the two around the middle genes. So where gene E and GB are, like when you write it, it doesn't matter.
Mhm.
EGB is the same thing as BGE.
Oh, okay.
Yeah, yeah, that was kind of like I don't know, like a trick question. Sorry, but
No,
uh,
you're good.
yeah, yeah. So as long as you understand also that that order doesn't matter specifically, I definitely would write it the first way, just because we've been looking at E on the left side so far.
Mhm.
But
So
cool.
okay, until like the actual like order is figured out, then as long as we put that G is in the middle
Yeah. Yeah.
at first. Okay, great. Thank you.
Yeah. Yeah, of course. Yeah, I hope that makes more sense. With matching, yeah, the double crossover genotypes with the parentals. So nice.
Yep. Sweet.
Thank you, Emil. You too. Yeah, of course, Brenda. Do you have any more questions about anything on the activity? Oh, no, she's gone. Or they're gone. Okay, I'm gonna pause my recording for now.